Today, we celebrate all the women of GRC who choose to challenge.
To those who are curious and committed. To those who asked questions.
To those who never gave up or took no for an answer.
Those who broke the glass ceiling and paved the way forward.
To those who served as leaders and trailblazers.
To those who served as mentors and advocates.
To those who have and will continue to shape the future. Here's to the women who have advanced the frontiers of science and helped make GRC what it is today. Happy International Women's Day.
Hello and welcome everyone to our second program in the GRC Connects Innovations series. Thank you for joining us from 35 countries and over 235 institutions as we celebrate International Women's Day. We are honored today to feature two members of GRC's governance, Christie Anseth, and our current board member, and Chad Mirkin, who served on GRC's Conference Evaluation Committee. Christie and Chad will speak about innovation by biomaterials. And I'm pleased to welcome Shana Kelly, who will introduce our speakers and serve as moderator. Shana is a university professor at the University of Toronto and a member of the departments of chemistry, biochemistry, pharmaceutical sciences, and biomedical engineering. Her research interests are in biophysical, bioanalytical chemistry, chemical biology and nanotechnology, and new methods for tracking molecular and cellular analytes with unprecedented sensitivity. She is an inventor on over 50 patents, a founder of 3 molecular diagnostic companies, associate editor for ACS Sensors, and an editorial advisory board member for the Journal of the American Chemical Society and ACS Chemical Biology. Shana, thank you for joining us, and I'll now turn the program over to you.
Thank you so much, Nancy. It is really a great pleasure to be moderating this session. Watching the video and all of those amazing women who have been involved with the Gordon Research Conferences over the years. It's a wonderful way to start this event. It's a great day, and we certainly have much to celebrate as a community. And so just before we start, a couple of housekeeping notes. So we will have Christie and Chad give their talks. There's going to be a short period after each talk where you can ask questions. Please use the Q&A interface to input your questions. You can do it during the talks. You don't have to wait till the end. And then after both talks conclude, we will have additional time for Q&A and discussion. So please don't be shy, put those questions forward or topics that you would like discussed, and we look forward to a really great session. So I am going to start by introducing Christie Anseth. And I have a very short bio here for Christie. One could go on and on about her many accomplishments. But let me just tell you a few things about her. She's a professor of chemical and biological engineering and an associate faculty director of the Biofrontiers Institute at the University of Colorado at Boulder. And she received the L'Oreal UNESCO Award for Women in Science in the Life Sciences in 2020. Her research interests lie at the interface between biology and engineering, where she designs new biomaterials for applications in drug delivery and regenerative medicine. So welcome, Christie. Can't wait to hear your talk. Please go ahead and share your slides, and we'll let you take it from here.
Wonderful. Thank you for the kind introduction. It's great to be part of this day and kind of raising my hand to celebrate all of the women out there. A very inspiring introduction. The Gordon Research Conferences have done so much to support and promote women in the sciences and engineering. And I'd like to maybe spend a few minutes, the half hour that I have, telling you some about what's inspired me in my career and working in this area and designing biomaterials and different types of material chemistries that can help promote tissue regeneration or also understand different biological processes by watching cells in vitro in environments that recapitulate what could happen in your body. And so all of this has been done at the University of Colorado with a great team of students and postdocs, and I hope to introduce you to some of them throughout this these few minutes that we have together. But maybe by way of an overview, here's kind of a high-level view of the way we think about biomaterials and the way that we might engineer them to coax cells to be able to promote regeneration or to study their processes. So we kind of look outside of the cell and we think about designing materials and different chemistries that can interface with those cells. And we really want to understand how we can introduce cues and understand outside-in signaling to the cell. And so over here on the right is one example of an application where we have coaxed chondrocytes that are found in cartilage tissue to regenerate cartilage, which is a tissue that has a very limited capacity to heal itself. And so that's been one of the areas in my field where we've had lots of successes in tissue engineering skin, regenerating cartilage, getting bones to heal faster. But oftentimes we don't understand some of the processes well enough when we think about more complex tissues and structures. So the smallest damage to the spinal cord, and we still don't understand how to get those nerves to regenerate. And so many times we go back to the material chemistry and we say there's some fundamental things that we can learn and develop new methods to understand this outside-in signaling, and we look to growing and developing cells and cell models of tissues in a dish. And so this can be very beneficial, not only from understanding biological processes, but more and more we're also looking at better ways to have models of tissues in a dish for drug screening and other applications. So I'll talk a little bit about this and some of the motivation behind this, but I thought I might begin with just a few questions. So some of this is anything that we do in vitro a good model of what happens in vivo? And hopefully I'll convince you that there are some great things that materials and biomaterials can provide in that direction. Another though is that as we seek to make more precise types of biomaterials and different ways to temporally and spatially control biomaterial chemistries, can we think about ways to be more precise with how we treat and design materials for medicine? So some of the advances in precision medicine, is that hype or hope? And is there future in that? And I hope to convince you of that as well. And then finally, I guess in this broader context of what role biomaterials might play, I think maybe the field could it be shifting towards designing one biomaterial that's designed to deliver a drug or treat a disease? Or do we begin to think about ways that we systematically and customize materials for individuals and designing them in ways to treat people for specific disease or with specific comorbidities or different types of treatments that would be personalized? And I guess in the very bottom here is a phrase that one of my collaborators, Leslie Lindwan, has introduced me to, is that a woman is not a small man, right? So when we think about this, oftentimes drugs are delivered to people based upon their weight or mass. And this is not necessarily true. There are lots of many differences between men and women. And so in this talk, I want to give you one specific example of how biomaterials can be used in this way, and we'll look at that with cardiac disease. But before I begin with this, there are many different types of biomaterials that one can use. And my own group has had a lot of interest in hydrogel materials. So here's a macroscopic picture of a hydrogel. If you could see the molecular level details of this hydrogel, you would see long chains of macromolecules that would normally dissolve in water, but because of the interactions between the chains, it renders them insoluble. And instead they imbibe large amounts of water. And that makes them very similar to many of the tissues in our body. It also makes them very good for embedding cells inside of these environments, because when you embed the cells in them, they can communicate with one another, and those molecules can be transmitted in a facile way because of the high water content. They have a lot of the mechanical properties of soft tissues. You can exogenously deliver molecules. And so as an engineer, sometimes we're very interested in these processes because we want to understand and manipulate and coordinate these complex events that occur on multiple time and size scales. So coordinating how tissues develop from the instant that you encapsulate cells in these environments to as they grow and recapitulate the mature tissue structure, but also the chemistry behind this, how you can look at things at a subcellular subcellular scale and how cells will bind and interact with molecules, to how cells can migrate and remodel through these tissues, to the larger scale of developing complex three-dimensional structures. And so oftentimes, though, in these hydrogel systems, you're not just interested, right, in putting these cells in these environments, but you're interested in how can I put in chemistry that the cells can bind to particular ligands within the hydrogel? How can it sequester molecules that can be delivered on a certain space and a certain time scale? Or how can the cells maybe even degrade or manipulate this environment? And so we oftentimes think about this as we go from kind of this general synthetic biomaterial environment. How can I introduce biological signals? And then sometimes I want to have an environment that it recapitulates what happens inside of the body, and it mimics many of the tissue environments, and the cells decide. So I'm an observer, and I'm trying to make something in a dish that looks like cells in your body. Other times I want to be an active experimenter, and I'd like to have something user dictated where I can exogenously manipulate the environment around these cells and then watch how they respond. And because of that, I learn something about how cells process these signals, and then I can design next generation of smarter types of biomaterials. All right, so we're going to begin there. And what I'd like to do is maybe say one of the ways that we introduce these chemistries is we take advantage of the advancements in different types of click reactions. So these are highly specific reactions, very efficient. They can occur in the complex biological milieu, and typically there are very few side products and very few side reactions is some of the definitions of the click reactions. And Carolyn Bertozzi was one of the first women in our field to introduce the concept of being able to do this in a biological system. So these click reactions, one of the first was this copper catalyzed azide alkyne reaction to form this triazole. But oftentimes these copper reactions aren't compatible with occurring in living systems. So what Carolyn did was she introduced us to some of these strain-promoted azide alkyne cycloaddition reactions, these SPACs. And here's one of the first examples of a bioclick reaction that allowed us to label all types of molecules in vivo. And we began to think about, could I make biomaterials out of these types of reactions? And now today we have many, many different types of diverse types of bioclick reactions. So the concept, our idea is, can I make different types of macromolecular precursors, such as that might be used to assemble a hydrogel? And if I choose different reaction partners and click them together, can I assemble these in the presence of tissues, cells, or biologics? And so in my group in particular, we've been really interested in these photo-initiated called thiolene reactions. So they're one of the few that can be light activated. And that provides some nice advantages because if I assemble hydrogels with cells and I put them on a microscope, then I can introduce light through lasers and different types of optics to change the chemistry surrounding a cells and really learn a lot about biomaterials design for tissue regeneration. So here's just one example. A lot of times we pick the thiol to be this amino acid cysteine that can be, you can use solid peptide synthesis to create many different amino acid sequences. And then if you just include the cysteine within your sequence, then you can react it with different types of synthetic hydrogel precursors, such as this PEG macromolecule here. And so the tricky part, or the key, is really which type of end group do you want to use? And here the ene is this ring strain norbornene. So what's interesting about this is when you mix these two together, they're very stable for long periods of time. But when you introduce light and initiate the reaction, these two will click together through a unique type of reaction. It's sort of a radical mediated step reaction. And you can create very highly idealized networks where you have a PEG component and a peptide component. And so now it's very typical and common in our labs that one can use liquid robotic handling systems, use arrays of different types of cell culture systems, and create hydrogels where you can introduce biological signals through the peptides, and you can control the connectivity and the properties of the hydrogel by the type of peg that you choose, its number of arms, its molecular weight, and print these into arrays on 96 well arrays and put it onto a high throughput imaging system. And you can look for different compositions that might be suitable to the types of properties and processes you're interested in controlling. So I want to put this in the context of one area that we've had a lot of interest in, and that's trying to understand how cardiac disease and fibrosis occurs. And the reason I chose this is looking back in hindsight, I got interested in this area and had some great collaborators and a great group of women that really contributed to this in my group. So long before the NIH started to talk about biological sex as something that we all needed to think about and care about, whether we're studying humans and disease or whether we're studying just cells in a dish, it's something to pay attention to. And so, women are known to have different mental health issues, suffer from more depression, also suffer from more osteoporosis. Cardiovascular risk is very different in men and women. And so in this latter one, I wanted to tell you a little bit about one disease that we've been studying and designing biomaterials for. And that is in your heart, you have 4 valves, and one of them is the aortic valve. And it can become very stiff and stenotic. And that can really impact the whole overall health of your heart. So here's one picture of the aortic valve. From an engineer's perspective, it serves as a very ideal one-way valve that directs flow throughout our bodies. But what can happen is over our lifetime, it can become diseased. And these valves, rather than opening and closing correctly, what happens to the cells inside of these valves is they get misregulated and they start to deposit and create a tissue that's very stiff and scar-like. And so then that influences the ability of these valves to function properly, and that can influence the overall heart health of your heart. And so here's one example of that. It can lead to hypertrophy or thickening or overgrowth within your left ventricle. And what's interesting about this is that men have a twofold risk associated with aortic valve disease, but women tend to be diagnosed less often. Men's valves also happen to calcify more, whereas women's valves tend to just become more scar-like and fibrotic. And right now, the main treatment is once you're diagnosed with this, unfortunately, there are no drugs available to slow or reverse this that are effective. So instead, you have your valves replaced. And this used to be an open heart surgery, but now there's even a minimally invasive technique called a transcatheter aortic valve replacement, where they go through a blood vessel and weave all the way up into your heart, and they can replace that valve. And what's interesting about this is replacing the valve is also known to kind of improve your overall prognosis and the overall heart health. So I want to talk a little bit about how biomaterials can help us in understanding this disease. So first, I want to say this is built on a great group of women throughout my field that have taken on this and trying to understand not just heart disease and valve disease, but also differences between men and women. And they truly inspired me getting into this field and interest in this area. And in particular, I collaborate with Leslie Lindwan, and Kristen Masters is a former postdoc in my group who's now a faculty member at Wisconsin. So it's a great community, and it's been a lot of fun to work in this field, and I've learned so much from them. And then within my group, in looking back historically, I also realized how many fantastic PhD students and postdocs came from my group. So here's a former PhD students. Leslie's a collaborator. Here's a current and very recent graduates who are still within the group, and a very talented postdoc. And then up here on the right is we also collaborate with people in our medical school. So we have access to data from patients that suffer from these types of diseases. So I'd like to tell you a little bit about some of what they've done. And so in looking back, so I told you that these are the aortic valves. They can become very thickened and diseased, and so they don't function very well. But if you could see some of the cellular level details, what you would see is we're looking at this one molecule here. It's called alpha smooth muscle actin. And what that really is a marker for is when you look at all the different cells within your heart valves, when they become activated and become a wound healing phenotype, they call them myofibroblasts. They express this protein, alpha smooth muscle actin. And in disease valves, for some reason, the cells get stuck in this wound healing state. So they become an activated myofibroblast that persists for a long time. And that's part of the problem, is that rather than being healthy and repairing the tissue, they get stuck in this state. And so we've been really interested in trying to understand this transition between a healthy cell population over here, where they're more of a fibroblast, to what happens when they become activated. And biomaterials have really allowed us to understand a lot of this. So Sharon Wong was a biology student jointly advised by myself and Leslie. And it was very early on when there was a first array that could be used to study these valve myofibroblasts and fibroblasts isolated from valve tissue. And one of the things that we found quite interesting is that before we even began our study was what we realized, that the minute you took the cells out of the tissue and you put it in standard culture dishes, which are made out of polystyrene, that the cells completely change their expression profile. So you don't need to know the details of this, but hopefully you can appreciate that there are lots of differences by simply putting these cells in vitro on a dish versus what they are in the tissue. And one of the problems is that when you put them on really stiff environments, they all become myofibroblasts. So it made it really hard to study these cells and what was causing this disease process. So one of the things that we did early on was Julie Benton, who also became an employee. She works at Medtronics now, and she's done a lot in designing new types of materials and devices, even involved in some of that transcatheter aortic valve replacement. And April Clarkson, who was designing, we really wanted to understand, could we design materials that would allow us to understand these valve cells and when they are quiescent and when they became activated, and maybe even what would cause them to calcify in some of the valve disease that happens frequently in men. And so here's just a picture of these on a dish. It looks like my media is not linked to, oh, it is, okay. So if I click on this video, what happens when you put these cells on a dish is that they all become activated, they highly contractile, and they snap, and they form these types of nodules. And these nodules eventually will even go on and calcify if you watch them long enough. So unfortunately, many of the standard culture techniques were much more like a disease in a dish. And we really wanted to understand and be able to reverse some of these processes. So this is where biomaterials came into play. And so what we did was we looked at these cells within a valve tissue, and we looked at them in standard tissue culture methods. And then we designed different types of click hydrogels where we could match some of the properties of the valve tissue. And we could control and make these cells, so in a dish, they become very activated. In our soft gels, they behave much more like they would in a valve section. And we could even look at the protein levels. Some of the details don't matter so much here, but the soft gels prevented some of this activation. And so then that made us really interested. So we did a lot of characterization and the molecular level features of these cells in these different biomaterial environments. And so here's just showing what happens. These are a bunch of different genes we can look at for these cells, and maybe just look at the patterns of this rather than necessarily looking at the details of what the genes are. But when the cells are within the tissue and freshly isolated, there's a certain pattern. And when they're put on plasticware, they're very, very different. And if we put them inside of our gels, it's not perfect, but it starts to begin to recapitulate some of the aspects of cells within the valve and cells that are just freshly taken out of the valve. And so we began to think about, well, maybe there's much about some of the mechanosensing that these cells have. And so at the time, we were designing different types of materials where we could intricately control the stiffness of these types of biomaterials. And we were able to in situ regulate it and change it. So we were really interested in how does matrix stiffness or elasticity affect this environment, and how does that change with disease? And would there be ways that you could reverse it? Because if you could think about some of this mechanosensing, maybe that might give some insight into new types of drugs or targets that could be druggable for treating this type of disease. And so this was the work of April Clarkson, who's a professor at the University of Delaware, and Andrea Casco, who's a professor at the UCLA. And they had designed some really clever chemistry where we could make hydrogels. And within the crosslinks of these hydrogels, we put in these linkers that when you expose them to light, they would undergo an intramolecular reaction that would cleave. So the materials were completely stable, but if I introduced a laser light from a confocal microscope, I could begin to cleave these crosslinks and soften the material. And so that's what they did. So if I exposed it to light, I could I could continually soften the material, start with a stiff material and soften and understand continuous changes in the cell microenvironment. Or I could soften it partially and then remove the cell source, and then I would have a material that was about half of its original stiffness. And so this was important or interesting to us because we could begin to understand what ranges would these cells activate and when might they deactivate. And so maybe just look at this red line here. We could design culture environments where in stiff environments, almost all the cells were activated in these myofibroblasts. And in the softer environment, only about tens of or less kilopascals, most of the cells were these quiescent fibroblasts. And so now this became very interesting because the unique experiment is to look at the reversibility of this. So one of the first things that we did, and this was also a collaboration with Chunyan Yang, who was a PhD student, and Lena Bastia, who was an undergraduate student that came for the summer to do research in our group. And so we were doing all kinds of experiments where we could culture cells in stiff environments and activate them. And then at any point in time, we could soften this and then watch how the cells would respond and would it be reversible. And so indeed, like we could culture these in environments where cells were activated, and if they're in soft environments, they were not activated. And then after 3 days, if I soften it, we could ask the question, would they deactivate? And on these short time scales, indeed, cells did deactivate. And so what's unique about this experiment is we can begin to ask questions about what cell signaling processes are involved and allow this reversibility. And so I won't go into the details here, but with our biology collaborators and some of Sharon's work, we could figure out what were some of the key pathways. And we began to screen through and look at different types of drugs that could allow this type of reversibility in the myofibroblast. But it's not always reversible. And so this is some of what happens in disease. And so Sierra Walker and Anouk Collars continue on in this study, and they looked at many, many different timescales. And as part of disease is what happens is there's an accumulation that cells are in these stiff microenvironments and irreversible changes occur. And indeed, they did show that depending upon how long cells are in these stiff microenvironments, if you change their environment at later points in an experiment, all of a sudden something irreversible happens. And rather than cells being able to reverse back and become quiescent, they'll persist and they will not revert. And so this inherently led us to understand there's probably something going on in the nucleus and different rearrangements and epigenetic changes that are occurring. And perhaps this is also part of the disease process. So they've done some really nice work in this area. Then that continuing on, I guess that made us think about, let's go back to this. All of this is in vitro work, and we're trying to understand things and do very controlled experiments. I mentioned to you that there are differences in sexes in how cardiac fibrosis presents itself and how valve disease progresses. So then we began to ask, does this link to anything that's observed in people and in patients' tissue? And so that's some of our more recent work that we've worked on. So this is Megan Schrader and Andrea Gonzalez Rodriguez, a couple of very talented PhD students who they did a lot. They characterized the valves from patients that had aortic valve stenosis and have shown things that here's male tissue and sections, and the black is their calcification that occurs. And the females do have calcification, but it tends to be very micro deposits. And so looking at some of these differences, and they also showed that if you stain for something like collagen that's found in your valves, that females that have AVS have lots and lots of collagen that gets very cross-linked and makes it very stiff, much more so than the male tissues. So in trying to understand this, they were able to design different biomaterial matrices that also recapitulated some of these aspects that are observed in humans. So we took both female cells and male cells and designed matrices that recapitulated some of their diseased environments. And indeed, we found that many of the male cells and male samples would calcify more heavily, and many of the females would have a much more robust and more fibrotic or collagen-rich type of matrix instead of calcification. So that's one of the directions that we're going, and we're using this with different types of array platforms to understand and screen through different types of combinations and molecules that might be able to treat this uniquely depending upon the progression of the disease. But in addition, I mentioned that the state of the art right now is to have this valve replacement surgery. So we've collaborated with a talented group down at our medical school, and Kate Schutze, she collected samples from patients that she was treating with this TAVR process, and she would collect blood serum prior to their procedure. And then one month later, she also collected their blood serum after the procedure, because there's some thought that you're not only replacing the valve, but it also improves overall heart function. And so we got these samples, these very precious samples from our medical collaborators. And then we had a team that we were looking at, well, what's different in these two blood samples? And if we could understand what's different about that, then maybe we could understand, does that have an impact on the whole heart, and design ways to maybe improve it even without a valve replacement surgery. And so again, the types of experiments we did is we had these great in vitro models that we could culture patients' heart cells in a dish and the valve cells, and we could expose them to the serum from the patients before they had this procedure and the serum after they had the procedure. And we had a collection of different types of patients, both males and females. And maybe the biggest point or takeaway is this graph here on the right, is that by doing these in vitro studies and training our data set, we could understand what was the role of the matrix environment and predictability that a patient would respond well to this TAVR treatment by looking at different measures of what happened what their aortic valve stenosis was like and their overall heart performance after the procedure. So we're beginning to think about ways to design and use biomaterials to personalize in predictive ways for what patients' outcomes might be after a procedure or surgery. So then that was just looking at their serum, and we could make predictions and correlations. But we also look at proteomics and what are the differences? What are the different molecules that are present pre and post TAVR? And so we're beginning to deconvolute and look at very specific ways and ask very defined hypotheses. And so here's just a few of, so we've identified at least 3 different targets, some of them related to inflammation and other processes in the heart. And we're able to understand what's happening and leading to some of these sex-specific differences within the heart and the heart valve. So I think I'm out of time. But I wanted to maybe just acknowledge and say, it's a privilege to work in a university environment. I get to advise a fantastic group of students and postdocs that are very talented, and my main goal is to help them succeed in their future career plans. They did every single experiment that I showed you today, and it's certainly a delight. I learn something new all the time. I've also had the pleasure of advising many fantastic women, and I'm really excited about the future, and it's great to be able to present and celebrate on this day. And so just briefly, I'll acknowledge the names here. I think I've highlighted everyone throughout, but we've also received very generous funding from the National Institutes of Health and NSF, and had a great group of collaborators. And with that, I'll stop sharing, and I would be delighted to answer questions.
Great. Thank you so much, Christie. That was a wonderful talk. We have some questions coming in, and I want to remind everybody who's online to please type in some questions into the Q&A interface, and we'll get to as many as we can now, and then we have more time later. So I'll start with some questions about the models, and there's a few questions here, so I'm just going to do them kind of one at a time. And so first of all, this individual, truly says, thank you for your wonderful talk. Could you please discuss how you chose the various disease models or biological systems to work on? So maybe we can just start with that.
Yeah. So, I mean, I think early on in my career, we sort of started at the musculoskeletal system where you could work with a single cell type, trying to understand, having a simplified model and understanding how cartilage might be able to regrow and regenerate, how bones which can naturally heal, how they could heal faster. And then in the example I gave today, it was sort of fortuitous. I was down the hall from a faculty member, Leslie Linewan, who was someone as a junior faculty member I truly admired, and I just enjoyed talking science with her. And she worked on diseases of the heart. So I thought, I need to think out some way to collaborate. And the heart valve was an area that seemed really intriguing. And it was one that when you isolate the cells from the heart valve, you really do need biomaterial matrices and environments, because all of the cells activate. So it seemed to be a great example and a synergy with where we could design biomaterial matrices to learn something about cardiac fibrosis. And we've been collaborating for about a decade, and I think the interest has just grown and grown and grown. So sometimes science is something that you think about and say, I'm going to plan this and find the right collaborators, but sometimes it's just fortuitous that you meet people and you learn about their areas and you think, oh, I have something that might really be helpful for this area, this field. And that's what happened with our interest in cardiac fibrosis.
Great. Yeah, that's a great story. I couldn't agree more that there's so many interesting problems for people like us to work on that have platforms and technologies that can be deployed, a variety of things. But it's those personal connections that you make and the people that you listen to what they're working on. You have a solution, and then an idea is born, and then you run with it. And I can think of so many examples of great work that's happened because of those kinds of interactions. So another question from Julie. So how do you design hydrogels to match the heterogeneity of tissue? So for example, material properties, but also differences in ECM that we see within the body.
Yeah, and so I mean, I guess my answer is kind of twofold with that. We do a lot of work with photochemistries, and especially with a lot of advanced fabrication technologies, one can make very advanced materials, sort of layer by layer. And like the one example I gave with us using peptides, it's fairly straightforward to introduce peptide sequences that are specific to a tissue and the sequences that are found in proteins within that tissue. But having said that, right, that hydrogels that we work with, they're cross-linked, they're amorphous, they don't have the fibular structure of tissues. And so sometimes we also think like how simple is complex enough? How much do you need to recapitulate the actual tissue? I mean, of course, if we could recapitulate everything about a heart valve tissue, then we could just assemble the cells and assemble the exact ECM, and you would have a heart valve, right? In addition, but we aren't there yet. And so it's sort of like taking advantage of how much can you rely on the cells to assemble and do this versus how much do you need the biomaterials to direct it. And I don't think there's a simple answer to that, but we kind of take both strategies sometimes.
Absolutely. Okay, so we have a comment and a question here from Ben Yang, who says, fantastic talk. Have you used your in vitro model for drug screening or discovery? And if so, can you just share your thoughts on the prospects and challenges associated with this kind of work?
Yeah, so absolutely. I think we do lots of screening for different types of inhibitors or drugs that might be useful for whether we're trying to differentiate a cell or reverse fibrosis, like the example I gave right now. I think some of the challenges that exist are, we don't always acknowledge it, but culturing cells in 3 dimensions or 4 dimensions, you're varying something in time. I think we all kind of say this is a better model, what's happening in the tissue, but all the assays become harder. All the imaging becomes harder. If you want to collect your cells afterwards and analyzing it becomes harder. So those are some of the challenges. So we're constantly looking at ways when I'm doing screening, what are some high throughput real-time ways that I can characterize the processes that I'm doing, because some of the assays are more difficult in 3D. But I think that what's interesting, at least to us in certain areas, are that if you screen on tissue cluster plasticware, that's very beneficial, and it's very, there's some things that are simplified, but I think you miss, and we see different drug targets in 2D versus 3D, which to me, I find that more examples like that are going to be forthcoming in the field, and it might be really helpful that things get overlooked early on and screened out early on because of that.
Yeah, absolutely. Okay, so this is a question or a topic from Yang Sun, who is asking how hydrogels, I think the question is, are they being used clinically? How are they being used clinically, and kind of what's coming around the bend?
Yeah, so I think for us, so some of the thiolene chemistries that I've mentioned, we've been working on those for almost a decade. And what I will say is that now So they've been licensed, and a company, Mosaic Biosciences, is translating them to clinical applications and using them to deliver drugs and other molecules more efficiently. But it takes a long time every time you develop a new chemistry. So again, there's a little bit of that gap. Do I use the off-the-shelf materials that are approved and use them in new ways versus designing new types of materials? And the new materials, I think, can be very impactful, but it can take a longer time. Where I think some of the more sophisticated chemistries, like some of the light cleavable and the light degradable materials, there might be niche applications in ophthalmics and things like that. But I think what's more valuable about those is, as we think about making better tissue models in a dish, there's a lot of chemistries that one can do to make better microphysiological systems in a dish that are going to be really useful for drug screening, understanding diseases, identifying new targets. So that's probably farther away from being used in humans.
Absolutely. Yeah, I mean, it's one thing to actually take something into humans, but there are so many other things that you can do with these kinds of materials that have a tremendous impact on human health. So no doubt about that. Okay, maybe one more kind of quick question. So this one is from Sophia Bailey, who is asking if you could comment on the compatibility of radical mediated chemistries for building hydrogel networks in the presence of proteins in cells.
Yeah, no, that's a great question too. So every time that we do these experiments with light and different initiators or radical mediated, there's always conditions where it becomes cytocompatible and physiologic that it can be used. Our cells have lots of natural defense mechanisms to protect us from, we walk outside and ultraviolet mediated damage. But there are also photodynamic therapies that they use lights and radicals and other chemicals to kill cancer. So it's all about finding the right conditions and processes. So yeah, it's definitely something that we're constantly looking at and defining which regions are compatible. Yeah.
Okay, great. All right, well, thank you so much, Christie. Thanks to everybody who brought questions forward. We are going to come back. We'll have Christie kind of disappear, but then she'll be back and we'll do some more discussion and Q&A at the end. So don't worry if your question didn't get answered on this round. We will get to it the next time. But now we're going to switch gears for the next talk. And we are very fortunate today to have Chad Mirkin with us from Northwestern University. So Chad, you can put your slides up and come on screen here when you have a second, and I'll just get going with your bio. Again, this is a very short and abbreviated bio for Chad. Chad is the director of the International Institute for Nanotechnology and the George B. Rathman Professor of Chemistry, Chemical and Biological Engineering, Biomedical Engineering, Materials Science and Engineering, as well as Medicine at Northwestern. He's a chemist and a world-renowned nanoscientist expert who is known for his discovery and development of spherical nucleic acids, which we refer to as SNAs, and SNA-based biodetection and therapeutic schemes. He is also the inventor of dip pen nanolithography and a series of related technologies that are cantilever-free nano patterning methodologies. He's also developed on-wire lithography, coaxial lithography, and he has made many different contributions to supramolecular chemistry as well as nanoparticle synthesis. So with that, Chad, we'll let you take it away.
Great. Thank you for the very nice introduction, and I want to thank Nancy for the invitation to be able to speak today and celebrate International Women's Day. Christie gave a great talk, a tough act to follow. I'm going to spend my 30 minutes talking about a few things and a few different themes. One is this topic of nanomedicine, and specifically why scale and chemical structure make a difference. And I think that will become clear by the end of the presentation. But I also want to use this to talk a little bit about some of the really important contributions that women have made to my group and really to the growth of Northwestern over the last 30 years. What I loved about this was the ability to kind of look back in a different way, just to take some time and have a historical look back and, ask the question, well, what were the significant contributions? How did women scientists really change both Northwestern and my group? And it's pretty interesting, at least it was for me and many of my members of my group who helped me put this presentation together. I've got on this first slide two people that really were instrumental in all of that. On the right is Elizabeth Singewald, who became a scientist at DuPont Central Research and Development and is now a professor at Newman University. She was actually my first PhD graduate student. She brought into the group an incredible work ethic, excitement for science, and just will to succeed that permeated the group and it really still does so today. It really set the stage for much of our success down the road. And I think without her, we would not be where we are today. And then on the left is Hillary Godwin, who used to be at Northwestern. We recruited her. She actually has an important part of history. I mean, Northwestern, unfortunately, had no women in chemistry until the 90s, which was just awful. It wasn't from a lack of trying, and I don't think it was because of environment, but there were so few. It was kind of representative at the time. There were so few women, and many, obviously, departments were recruiting them. She took a chance and came to Northwestern, and it turned out to be just a fantastic situation for both parties. And she also completely changed the chemistry department, the culture, and did it in a really positive way, in a way that led to this explosive growth that I think many people have seen Northwestern undergo, and what we're very, very proud about. She's actually now a dean at the University of Washington in the School of Public Health. I want to talk about nanotech, and in particular why scale really makes a difference. Just because something's small or big doesn't make it better or worse. But the nanotech opportunity in terms of medicine, I think, is really interesting. We often think of nanoscale structures as being tiny, but of course, I'm a chemist, and I think many people in the audience are chemists. From a chemist perspective, nanostructures are actually quite large, and that length scale is really quite interesting for a variety of reasons. One is, of course, we know that when you take and miniaturize bulk materials to the nanoscale, they have new properties, and we can take advantage of those properties. That's just where it starts. The other is that when you get down to this scale, you can begin to get structures that interact with biological systems on their length scale and can do things that small molecule systems can't do. In addition, when you can work on the nanometer length scale, you can begin to build architectures that have a type of multifunctionality that you just can't realize with small molecule-based systems. So that large size, from my perspective, is in large part what makes nanostructures for medicine so interesting. The ability to build multifunctionality into systems. That's going to be really important with respect to the theme of today's talk. And then finally, with my chemist hat on, the fact that these are so modular means that you can begin to systematically adjust structure and create structure-function relationships and understandings that are really quite meaningful in terms of not just leading to something that's good, but leading to a set of rules or design considerations that allow you to design into the future. And we'll talk about that as well. So this actually was covered a little bit in the introduction. If you look back, I started 30 years ago. And so the group is, I think, just going to be 30 years old very soon. And over the course of that time period, a lot of really interesting discoveries have been made in the area of nanoscience that center on this issue of synthesis and modularity. Spherical nucleic acids were mentioned. We'll talk about why those structures are so interesting, why they define a new class of nucleic acid. DipPen, of course, which was a tool for patterning that became a commercial tool used all over the world, still is used today, for making nanostructures and looking at the consequences of miniaturization on surfaces. And then we began to understand the properties of spherical nucleic acids and how they could be used in the development of diagnostic tools and then ultimately therapeutic tools. And we began to move towards this issue of shape control. And so we had to develop the chemistry to control particle shape and now can do so almost with complete control over a geometric shape. Began to think about how you can do nanolithography not on flat substrates but on rods themselves, nanorods themselves. That was the birth of things like on-wire lithography. And then as we kept moving forward, we kept asking, how can we develop useful technologies out of this? That led to things like nanoflares. I'll talk a little bit about those architectures and how they became the first tool to actually measure the intracellular content of mRNA in cells, and provided a lot of interesting capabilities, both on the research and medical diagnostic side of things. We eventually got rid of scanning probe, or not scanning probe instruments, but AFMs for dip pen. And now we use cantilever-free techniques, which are the basis for polymer pen and beam pen lithography. In doing so, we transitioned those types of tools from single pen tools to tools that now use hundreds of thousands of tips, many of which are independently controllable of one another. So that you not only can pattern surfaces, but now we've come full circle and we've begun to use these tools and think about these tools as chemical synthesis tools, where we can begin to synthesize material libraries that are massive. So this is done at scale, no longer just one at a time, but millions at a time, and where you can begin to explore the material's genome like it's never been explored before. And then most recently, we've moved heavily into the development of tools and materials, nanomaterials that can be used to impact the development of real medicines. And in fact, we have 6 drugs now in the clinic, in human use, that are showing a lot of promise, spanning many different diseases. And I'm going to introduce the concept of rational vaccinology. Some of you may have not heard about that before, but it's something you're going to hear about, I think, in a big way, because it really highlights what you can do on this scale that you can't do with molecular systems. And I also want to share a current example of a technology called FitFlare technology that's really quite exciting and takes the nanoflare concept, I think, to a whole nother level. Okay, so spherical nucleic acids, what are these? As I said, they're structures that are made by taking particles, nanoparticles, made of many different materials. In this case, I have a liposome core. Most people put things inside liposomes. Once we understood the spherical nucleic acid architecture was a privileged architecture, had unique properties, we began to think about different ways of making it. And one way was to take linear nucleic acids with hydrophobic anchoring groups, cholesterol, tocopherol, and insert them into a liposomal core. And at high density, you can force them to stand upright, and now they adopt the central shape of the particle core. Why are these so interesting? Well, they exhibit properties that we haven't seen before. First, they're highly biocompatible. If you're going to use them as medicines, that's going to be important. They, unlike DNA and RNA, naturally enter cells rapidly and in high quantities. We understand why that is. There are structures called scavenger receptors that can glom onto the clusters of oligonucleotides, they have a high affinity for them, and facilitate a process called caveolin-mediated endocytosis to internalize them. The restructuring of the nucleic acids, remember there's a 3' and 5' end, and the right orientation can prepare these types of architectures to engage a complement. And you're going to see not just a complementary strand of DNA, but also a complementary target in a way that leads to higher affinity binding. And that's kind of important because if you're going to use these as probes, you'd like to be able to soak up low concentrations of target. That allows you to do that and exceed what you can do with linear nucleic acids. The dense packing leads to increased stability. If they're going to be in a biological milieu, that's important because nucleases and other types of cellular machinery degrade nucleic acids, and that leads to then a longer lifetime in those environments. And then the final thing is any particle can effectively be a core, and we've made now hundreds of different versions of these types of structures. And that allows you to modulate both the chemical and physical properties of spherical nucleic acid. Here's the first example, and it's kind of an interesting one. This goes back now quite some time. We began to use gold versions of these architectures as probes, and the idea was to create a high sensitivity array-based detection platform. It's called the scanometric detection platform. And the way it works is you basically have a microchip of DNA, and you have particles that have been designed to recognize target DNA strands in solution. So from a sample that's been processed to release that DNA. Under the appropriate conditions, the particles will bind, the high binding constant will lead to a sequestration of the target. And then the chip will isolate on a particular spot that localizes it to that particular area and allows you to visualize it, because the gold particles are highly colored. Nano gold, of course, is red in color, and its intense color is one way to observe this. But the other property that nano gold has is that it's catalytic with respect to the reduction of silver 1 in the presence of hydroquinone. So if you flow ordinary photographic developing solution over this chip, anywhere where there are gold particles, you plate out silver, and you can increase the signal by orders of magnitude in a matter of minutes. And so this becomes a really high sensitivity and very selective way to detect different targets associated with disease. And that's pretty exciting. That's led to a commercial system called the Verigene system, which is now sold by Luminex all over the world. In fact, it was used even for COVID testing, but used for identifying lots of different targets associated with bacterial infections, viral infections, cardiac tests, human genetic tests, and pharmacogenetic tests. It's kind of an example of early technology in this case, ultimately going through refinement, engineering, and ultimately finding widespread use, and will continue to find widespread use. It was one of the first point-of-care medical diagnostic tools of the modern age of nanotechnology, developed and widely adopted and used around the world. Okay, a second structure that came out of this was something called the nanoflare. And there were several people involved in developing this technology. Dwight Safaros up at Toronto, David Giljohan, who's now at a company called Execure, but also this woman, Dan Zhang. They got together and they worked as a team to develop nanoflare technology, which was pretty interesting here. You can see we have an architecture that in cartoon fashion has a gold particle in the core. It has the oligonucleotides that define a spherical nucleic acid. But then it has a short oligonucleotide with a fluorophore hybridized to it, and that fluorophore is near the gold surface, and that's quenched. And it's designed to be that way, because the idea behind a nanoflare is that if you expose cells to these structures, they'll be taken up like spherical nucleic acids. And once inside the cell, if they can engage RNA, they will engage it selectively if you designed this appropriately, because there's a sticky end that can bind. And thermodynamically, binding of the target mRNA is favored because it's a longer overlap region, and you peel off the flare, turning on fluorescence. And over here, you can see nanoflares actually working. On the left, you can see breast cancer cells treated with nanoflares designed to go after a gene called survivin, mRNA called survivin, that's elevated in breast cancer cells. And you can see, just even with the naked eye, the fluorescence is quite intense. If we use the wrong type of flare, you still see some fluorescence. I'm going to come back to why that's there and why that's important in a little bit. So you have background, but you can very clearly see the difference between these two. In fact, you can set thresholds now and use flow cytometry to begin to sort cells based upon differences in genetic content. And so we began to develop all sorts of tools based upon flares. In fact, Millipore produced 1,600 different versions of these structures, and then they're used all over the world, even to this day. And they're used for looking at rare cells, for example, isolation of circulating tumor cells based upon differences in genetic content. They can also, though, be used for high throughput drug screening, looking at the up and down regulation of a particular gene, particular mRNA fragment. And people have even used them. A group in Singapore, Chenji, took this type of structure and began to utilize it to look for markers associated with abnormal scarring in rabbit models. And these were the first uses of nanoflares in living systems, in animals. And so kind of a neat example of a pretty simple capability that comes from this observation that structure makes a difference in terms of cellular internalization. That's the good news. The bad news is the following. These are not perfect structures. They've got some flaws. So lifetime's important, right? I don't mean just fluorescence lifetime. I mean lifetime of the construct is important. So if any degradation occurs, you release the flare, and that leads to background. That's why we have background in the system that should not be turning on. They're kinetically challenged because this is inherently a displacement assay. So it'd be nice to get rid of that challenge. Oops. There we go. they are taken up via this endosomal pathway. So the bulk of them are initially in endosomes, and then a small amount is released into the cytoplasm. That's your active agent. So you're kind of fighting that as well. And so a lot of kind of a trial and error and manipulation has to occur to actually get them to function properly. But at the end of the day, you know they're functioning properly because you can detect in a sequence-specific manner the RNA, which is actually in the cytoplasm. So the question is, how do we make them better? And that's where Devlina comes in, along with another student named Sasha. They began to think about this and said, wouldn't it be great if we could have a simpler design, if we could avoid false positive signals, if we could get a large amount of signal, if we could create structures that were better than FRET-based type sensors that we contemplated making, and that avoided this displacement problem, where we could make them kinetically faster. And also, wouldn't it be great if we could not just go after nucleic acids, but ions, proteins, and small molecules? Well, that's kind of a tall order, but in the group we've been accustomed to going after big challenges. And so you can see what we came up with was this forced intercalation strategy, where instead of having a nucleic acid recognizing target and having that binding event ultimately lead to a displacement event that turned on fluorescence, we would take a fluorophore that we could attach to an oligonucleotide that would be spectroscopically silent, in other words, not fluorescent, until it engaged in a recognition event that led to duplex formation, where it could intercalate, restrict rotation, and turn on fluorescence. And so that's pretty neat, because if you began to think about merging that with the spherical nucleic acid type concept, you could have something quite powerful in principle. And I'm not going to show you all the results. They've done a tremendous amount in this particular area, and I think it's pretty exciting. They're really poised to develop tools that can span really all classes of analytes. But here's an example. We worked with the Air Force, Peter Morrell and his group at Wright Pat, showing that you could design aptamers with fluorophores in specific positions, designed in the one state to be non-fluorescent because they cannot intercalate into any of the duplex regions. But upon target binding, they clamp down onto the target, form a duplex region now that is in close enough proximity that they can, a fluorophore can intercalate and turn on in terms of fluorescence. And in this particular example, you can see we've developed a detection system for DHEAS, a steroid that is associated with stress. And that's they're part of the human performance ring. That's what they're very much interested in. I think this is pretty exciting, because now you can think about developing these types of constructs for a wide class of different types of targets, and we have, in fact, all of the different analytes that I shared with you. But we didn't want to stop there. We thought about this and said, hey, this is great, because we can now begin to think about using other types of cores as well. We don't have to use gold anymore. We don't need a quencher. So we can swap out the gold with other functional entities, even proteins. And so now we've come up with pro-SNA type architectures, where the nucleic acid shell enables a nucleic acid-based analyte recognition and facilitates cellular uptake. That's all key, part of the whole SNA story. And also imparts nuclease and protease resistance, right? Because if we use a functional protein, that's going to be important as well. We definitely want to inhibit nucleic acid degradation, but we'd also like to inhibit protein degradation as well. And the protein core could allow us to enable protein-based analyte recognition, allow for really high target specificity. And if we use enzymes, we could begin to create amplified forms of sensing as well, all of which would be very attractive. And so the question is, does this work, and how would you think about doing it? So here's a simple example. Let's say we wanted to be able to measure the intracellular pH of a cell. Well, we can take eye-motif architectures, which are known to be pH sensitive, undergo this proton-induced folding type of event. And in that process creates duplex regions that allow for intercalation. So by synthesizing these structures with dyes, again, that in the non-intercalated state are off, but in the intercalated state turn on, you can begin to, in principle, map out differences in pH. And so the range at which this transition takes place is between pH 7.5 and pH 5.5. Well, here you can see an example of that. These are cells. This is an intracellular pH differences being measured, going from pH 5.5 to 7.5. They're clamped at those two pH values. Okay, what about other more exotic capabilities? Well, we can take really almost any sort of protein, and if we can functionalize it appropriately without degrading activity, so for example, glucose oxidase, we could turn that into a glucose sensor in principle. And so they did that. This is pretty exciting. All this very recent data, you can take and modify glucose oxidase with the appropriate oligonucleotides, turn it into a pro-SNA. And now you can treat cells with these glucose oxidase SNAs, but also with a dye precursor, one that again is spectroscopically silent, but gets turned on if the glucose oxidase turns over the oxidation of glucose to generate gluconic acid and H2O2, peroxide. So the peroxide is the trigger here that then drives this conversion and turns the fluorophore precursor into the fluorescent state, fluorescein in this particular case. And so that's pretty interesting. Does it work? Here you can see it actually does. This is the in vitro example of that. So that's not intracellular, but greater than 120-fold fluorescence enhancement and a limited detection of about 5 micromolar. That's pretty good. It's very selective. Oops. You can see glucose here giving a signal. All these other entities, including even glucose 6-phosphate, very, very little signal. And it actually works in cellular systems. And so this is kind of interesting. This we think is really going to blow this wide open and allow us now to begin to design lots of different types of probes for different types of targets of interest. Okay, so that's detection. As I said, one of the exciting things that we've been working on over the last several years is this idea of now pushing these types of understandings into the development of new medicines. And so I've talked about this in the past. So I'm not going to spend a lot of time talking about it. Spherical nucleic acids, they're in the clinic already now, and there are many shots on goal and many tests that are being evaluated. Amy Powler, a colleague of mine, head of dermatology at Northwestern, was really instrumental in this. She saw us working on this and said, hey, it'd be great if you could put these in creams, apply them to skin, and knock down inflammatory markers. And sure enough, you can. They become great knockdown agents. You can use either antisense or siRNA pathways, and you can knock down inflammatory markers, in this case TNF alpha, but we've also done IL-17 as well. And we've taken them through animal models and also into the clinic in humans, where we not only can see knockdown in skin, very first time ever for any nucleic acid drug, but also we can reverse the effects of psoriasis. That's exciting, not just because of psoriasis, but there are over 200 diseases of the skin with a known genetic basis. And so this becomes a modular architecture to create a lot more shots on goal. There's a company called Execure that we started that actually is developing that platform and going down multiple paths with that capability. The even more exciting example is shown here to the right, and Priya Kamthakar, who's a oncologist at Northwestern, along with Alex Stagg, taken some early examples of what we've done in the lab at Northwestern in vitro and in small animals, where we saw promise that these types of constructs, when systemically administered, will do something that most constructs won't. They'll cross the blood-brain barrier and localize not only in the brain, but in the tumors, at least of animals that have implanted tumors, and that we can get knocked down. And in the case of a disease like GBM, a deadly form of brain cancer, we can significantly increase animal survival. That's exciting for a variety of reasons. One is the fact that you can systemically administer them and cross barriers like the BBB, but you can begin to now tackle really tough diseases in principle, like GBM, and maybe other types of neurodegenerative diseases as well. This is now in a human trial. It was just finished. In fact, on Wednesday, I'm not part of the paper because I have a conflict of interest. The trial results are going to be published in Science Translational Medicine, and they look really, really promising, which is very, very exciting. They reflect a lot of what we saw in the small animal studies in humans. Okay, the last thing I want to talk about is this concept of rational vaccinology. Remember, I said that nanostructures are modular, and that's what makes them very attractive in terms of structure-function relationships. Well, this is very timely given what we're going through with COVID. I mean, the spotlight on vaccine development has been really important. We weren't developing vaccines for COVID. We actually are now, but we were developing them for cancer, in particular immunotherapeutic agents for cancer. And we began to look at what's been done in the whole vaccine arena. And it's somewhat troubling because, at least from a chemist perspective, because we like to think of the conventional way of making vaccines as the blender approach. People spend a lot of time thinking about the adjuvant, the immune stimulator, and the antigen, the immune system trainer. They spend very little time talking about structure. And from a chemist perspective, it's disappointing because I think we'd like to think that the chemistry matters and structural presentation is going to matter. And I would argue, where in the history of drug development has that not been the case? But in vaccine development, very little attention given to that issue. So we began to look at this and ask the question, is there a case for rational vaccinology, where it's not just the structure you put together, but the presentation of the components within the structure that matters, that architecture is going to be important. And the beautiful thing about this is we could use the SNA platform to kind of test that hypothesis. And that's what we've gone on to do. And so for those of you that aren't familiar with how vaccines work, as I said, there are two components, basically, an antigen and an adjuvant. Dendritic cells take these up, so getting them into the right cell type is critical. That's why putting them together might be really important if you think about it. And then what happens is you generate signals on the surface. The antigen gets processed and put on the surface of the cells. The dendritic cell gets stimulated. T cells dock onto these types of structures, and that's important because they then get trained to go out and find cells. Find cells that have that particular antigen and then effectively lyse them. So you're effectively training, in this case, the immune system to find cancer cells that have that specific antigen. And that's the basic principle behind vaccine development. Why would the spherical nucleic acids be attractive? Well, we know the sequences that are good adjuvants. They engage TLRs, and by the way, they engage them in the endosome, which is where these structures initially get taken up. So we thought we had a great hypothesis there that simply taking a linear nucleic acid and turning it into spherical nucleic acid would lead to a more potent adjuvant. That turns out to be the case. The second is we can begin to put the antigens, the peptides or the proteins that give this selectivity in multiple spots within the SNA architecture, in the core or on the surface, or even hybridized to the oligonucleotides that decorate the liposomal core. The other issues are, we know that these drain to the lymph nodes. They just like other cells, they're uptaken by dendritic cells without transfection reagents. They last longer, greater nuclease resistance, and we hypothesize that they would have higher affinity because we've pre-oriented the oligonucleotides to maximize affinity for TLR engagement. So now we were off to the races. But here's the problem. As I said, this is both an opportunity and a challenge. Even with a well-defined spherical nucleic acid architecture like the one shown here, if you look at all the different parameters, there are an enormous number that you can vary. So 6 features with 2 values and 5 features with 3 values, you end up having about 15,000 different SNAs that you have to sort through. So we've collaborated with Milan Mirkin to begin to use high throughput screening tools to look at functional properties that might make a difference. The core properties, the lipid composition, the size, for example, antigen properties, the type of antigen, the antigen location, the antigen density. And then you've got all the different oligonucleotide properties, which are many. Remember, there are many different forms. You've got the different types of engagers of TLRs. You've got the different anchoring groups. You could use cholesterols. You could use lipids themselves. Conjugation chemistry and orientation chemistry all can be important. PS versus PO, oligonucleotide density, and even complement density. And so we've begun to sort through those. And I just want to give you a few examples that are really quite exciting here. So here are 3 examples of classes of structures. We call them encapsulated, where the antigen's in the core, the oligonucleotide adjuvant is on the surface, anchored, and hybridized. Anchored has the oligonucleotides interspersed with the peptides on the surface of the liposomal core, and hybridized has an antigen that has an oligonucleotide complementary to the adjuvant and bound that way. We make these three structures so they have the exact same amount of adjuvant and the exact same amount of antigen. So this is the test of, does structure make a difference, or is it just the components? And we compare it then just to the components, the admix. Now, all of these can keep everything together. These guys, of course, the biodistribution is controlled by what they encounter. And here we've got three fantastic scientists, Cassidy Coleman, Michelle Teplinsky, and Shuya Wang, who really drove a lot of the results that I'm going to talk about here, and a lot of the work that I think is really fundamental in how we think about vaccine development. Because if you look at these plots, this is for cervical cancer, lymphoma, prostate cancer, you can see how the different vaccines retard the growth. And to make a long story short, if you look carefully, what you'll find is the hybridized structure is the privileged architecture. It consistently retards tumor growth and extends animal survival over all the others, including the admix. In fact, the admix is terrible compared to any SNA architecture. The first thing you learn is by putting them all together, you get an advantage. And by systematically changing where the antigen is and how it's presented, you get marked differences in terms of vaccine performance. And this one in particular is amazing because you're effectively getting almost a complete cure for a lot of the animals. So we then began to ask, can we go further with this? And so the neat thing about this is it opens up a type of personalized medicine that again would be almost impossible to envision via other types of approaches, where we're beginning to think about, can we take the antigens from tumor cells of a patient by taking those cells, lysing them, collecting the tumor lysates, and then encapsulating into the spherical nucleic acid core. Not the ideal location yet. We actually think we can improve on this by learning how to control location, and then re-injecting, using these effectively as a vaccine, a personalized vaccine. Now, we're not in humans yet with this, but we are in animals, and Cassie's done a beautiful job of tracking this, and you can actually see right here, they've created a construct for a really tough disease, triple negative breast cancer. And I have a friend whose wife has this disease, and he actually funded a lot of this research. Very, very exciting, because there hasn't been a whole lot done to create much promise here. What you can see is she's figured out that by taking oxidized lysates, and the oxidation is actually what makes them more immunogenic, you actually get tremendous suppression of tumor growth. It's completely eliminated in 7 of the 9 mice at day 20 in the vaccine. Compare that to untreated, the lysate SNA, so that's without the oxidized form, and then the oxidized lysate DNA, that's the admix right here. Marked difference, both in terms of suppressing tumor growth, but also look at the animal survival curves. So the way you prepare the lysates and the way they're packaged, this modularity is really critical in terms of controlling vaccine performance. Okay, so I just want to finish then with a last thought here. So looking towards the future. When we first started this, you go back 20 years, I think people were pretty big skeptics about nanomedicine. There still might be skeptics, but it's clear that we've made the transition from talking about possibilities to having real technologies that make a difference. You look at the COVID era and look at the shots on goal and the success of the Moderna vaccine. That is an example of a nanotech-enabled vaccine. And so I think the future is really bright in terms of multiple areas. Targeted drug delivery. I think nanostructures are going to be the key. This modularity and the ability to deliver multiple components in one shot. Very important in terms of getting tissue access, but getting into the cells and generating the signals when you want them to be generated. Injectable and wearable electronics. I didn't mention much about that, but of course we've got guys like John Rogers at Northwestern and other folks around the world that are just doing fantastic things in that area. I think lots are going to come out of that. Engineering immunity. I think it's going to be a big topic going forward, and lots of really cool things where, again, don't just kind of make a phenomenological mixes, but systematically design structures and ask a serious structure function type questions. And then finally, I think from a nucleic acid medicine standpoint, we've seen the wave of small molecules move to the wave of biologics. antibodies, I'm sure we're going to see a major wave in nucleic acid medicines. And I believe nanostructures are going to play a big role in making potent nucleic acid medicines, but also getting them to the tissues and cells that ultimately matter. Let me finish by acknowledging the people that have done the work. They're shown here. I mentioned some of the key people. I apologize for anybody I left out, but we have just an incredible group of people. Going back to that first slide, as I said, every one of them incredibly motivated and in this case, incredibly diverse. They come from 13 different countries. All kind of motivated by the common purpose of trying to push back the frontiers of science, ask serious questions and answer them, and have a lot of fun while you're doing it. And we've been fortunate to have a lot of different funding groups support our ideas and efforts. And I just want to make one call. I know we got a lot of people on the line. We have postdoctoral positions available for people that are similarly motivated. I'll stop there and be happy to answer any questions.
Right. Thank you, Chad. Amazing talk. Really incredible recent progress. We have a question here from Dolores Rose, who says, very interesting work. How do you manipulate the components so that you can tailor them to one disease versus another disease?
Good, very good question. Okay, so two issues when doing that. One is, what tissue do you have to get into and what cells? So, I gave you the examples of skin diseases. If you want to localize in the skin, you use creams. In the immune system, we're locally injecting and training the immune system locally, right? But then getting a system-wide response via the lymphatic system. But then the other part of this is sequence. Right? The oligonucleotide specificity, the targets of interest. So it's a combination of taking advantage of the characteristics of the spherical nucleic acid platform these pathways you want to engage with the choice of oligonucleotide, or I guess you could do peptide as well.
All right, great. A similar question for you, Chad. So, I mean, we think a lot about this just in the context of autologous cell therapies. So, in your last example where you were talking about making a tumor vaccine, I would imagine that you need tumor from each patient to do that. Have you thought at all about the logistics of kind of scaling that up and decentralizing it? Or do you think eventually there could be one source of the right antigens?
Can you hear me?
Yeah, can you?
You kind of went in and out there.
Okay, sorry.
I think I got your question. You were asking about thought about how you would even do a trial with this personalized type of approach.
Exactly.
So we've actually we have a very strong cancer center at Northwestern, and we've engaged the breast cancer experts. And Cassie and I have been beating on them to come up with first, is this a credible path forward or not? They think it is. And trials have been done with lysates, small scale, but it is the ultimate in personalized medicine, right? But as you point out, it creates both a cost challenge and an implementation challenge. And so, these are very early days, but I can tell you, we're going to do some of it. We're going to go down that path and see if it works, because the data are really, really promising. I mean, I don't think either of us, maybe Cassie did, she's more optimistic than I am, ever expected to see that type of success in that aggressive of a tumor in animal model.
Yeah, absolutely. Well, and it has been done, right? In some of the cell therapies where you're taking cells out of a patient and then genetically engineering them or doing something to them and putting them back into a patient, it's certainly been done.
Well, CAR-T in particular is a good example of that, but in this case is another added challenge, which is you're actually making the construct yeah. with what you've taken out and then putting it back. So we've kind of added another layer of difficulty, but yeah, that's why we're at the university, right?
Yeah, it can be done. It doesn't sound like the hardest problem you've ever solved to me, so I think it's very doable.
Oh, right.
Yeah, I was also wondering if you could say a few words about, I mean, it sounds like there's some really exciting news coming on the glioblastoma work with the the RNAs, the spherical nucleic acids there. Can you say a little bit just about blood-brain barrier? How do those things get through? Do you guys know what the mechanism of uptake is?
Yeah, it's impossible to really prove, but we do have models, the best models that are out there. Alex Steg has done a lot of this work that suggests that scavenger receptors are really important in terms of bringing in. So the fact that they're internalized and recognized by scavenger receptors is a clue, I think, that this could have happened. The models recapitulate that, but whether that truly is the reason in the living system, you put so many blinders on as soon as you go in vivo. Sure.
Yeah, of course. Okay, maybe one other question here before we bring back Christie. What are this is a question from Hen Hu, who says, what could be the possible reasons why hybridized DNA outperforms single-stranded DNA in the SNA-based vaccines?
Great question. So there's a paper called, I think it's called Rational Vaccinology. It was kind of first use of that term. It was published in PNAS. Shuya Wang mapped that out and looked at the kinetics of signaling. And it turns out that hybridized strain, and it makes sense if you think about it, right? So it makes sense, first of all, that putting everything together is important. If you need both in the same cell, having them together makes sense. Second is how they're processed and how long it takes, the chemistry behind taking the oligonucleotide off, taking the peptide off, and processing it is going to affect the timing of signaling. And she mapped that all out, and it turned out the signaling events were in sync with the hybridized structure, but out of sync. In fact, one of the main conclusions was of that paper, structure makes a difference, but timing may be everything. Because if that's true, now you can design not just spherical nucleic acid architectures, but other architectures that map on the optimum timing. So in other words, that we look at the spherical nucleic acid as a platform for kind of understanding those types of events and the importance of kinetics or lack thereof, and then use that information across many different platforms. So that's what I want people to understand. We don't just look at this, oh, it's a magical spherical nucleic acid, and this is what, it just happens to work. We're spending a lot of time trying to figure that out. And so now I've got another group of people that are making linkers where they can control the chemistry of release of the peptide, for example, and the timing of that particular event to see if we can move the signaling in and out of phase of one another to figure out what the optimum rollout is.
Wow, very cool. All right. Christie, if you want to come back, I'll just ask this one last question to Chad. What about using inorganic nanoparticles for drug delivery versus macromolecular polymeric carriers? So that's,
well, it's kind of interesting. The only example of where we used an inorganic particle, my inorganic colleagues would kill me for saying this, for therapeutics, is with the GBM case. And that was by design. That was the first one of the early ones. We developed all of our understanding of gold. But it made sense to do that trial with that construct. So one, we could learn how they worked, but two, so we could track them. So we could actually measure gold content in the tumors that were resected so that we knew how much of the drug actually got to the tumor. So it was a built-in diagnostic and therapeutic in one construct. Now that said, the vast majority of spherical nucleic acids going forward are ones that are made with biocompatible or benign cores. Not that the inorganic ones are necessarily harmful, but we just don't know. And so the lower hurdle, the lower bar, is to take a liposome, which has been used in lots of different types of purposes in vivo, and turn those types of architectures into spherical nucleic acids. So most of the trial data coming out of the commercial arm of this involves the liposomal structures. And you could easily convert, for example, the gold-based one into a liposomal one now that you know how they track, because it's the architecture that takes you to the brain, the outside architecture, not the core, that matters.
Great. Okay. Wonderful. So we have a bunch more questions, and I want to encourage everybody who's online, keep them coming. We've got some time here. We have about 15 minutes, and what I would propose is that we spend about maybe 10 minutes on kind of the technical scientific questions. Maybe we'll focus on the ones that are kind of big picture. And then I want to leave the last 5 minutes just to talk a little bit more about women in science and just your perspectives on, how far we've come to date and now where we need to go. So we'll save that for the last few minutes, just recognition of the day on which we're having this event. But let's go back to some science for a few more minutes. So another question for you, Christie. I had the same exact question. So, I mean, the differences that you showed for what was going on with valves from male patients, female patients, I mean, those differences were so striking. And so what's known about the underlying physiological drivers of those differences? Is it hormonal, or, you know, do we even know?
Yeah, no, I mean, it's a great question. It's complex. It's like the easy answer is to think just hormonal, and I'm sure that contributes some, but I think it absolutely goes beyond that. I think it's in the context of inflammatory events and overall health, and I think it's an accumulation, right? It's not, you know, it's not just a single point in time, but I think it integrates over time, and there are many differences in men and women. So that's what we're trying to understand. And I think I'm trying to look at the question too, but that I think the microenvironment synergizes with the secreted factors. But signaling is very different depending upon the local environment. You know, so
Yeah, sure. Yeah, I mean, it's a very complex set of things to figure out.
I think that's why we don't have any drugs to treat it.
Right. Absolutely. Yeah. All right. So back over to you, Chad, for a question. I mean, I love your comments about vaccines and how they've been developed to date. And I mean, honestly, I feel like until very recently, there just wasn't a lot of cutting edge technology being deployed, right, to develop new vaccines. It was kind of like we forgot why they were so important or why we might need vaccines on a much more accelerated timeline, but now we're all very aware of that. So I can see this area really exploding, and having people like you bring this molecular logic to it, I think it is just paradigm shifting. I'm just wondering if you could talk a little bit more about your perspective on that. You know, historically, why were we stuck in these very traditional paradigms, and you know, what does it mean to have this new, very modular way to engineer vaccines?
I think it's just some of it's just evolutionary. I mean, look, this has not been driven by chemists typically. So this will Christie won't like this. So much of vaccine development has been done primarily by medical folks and engineers, result oriented, right? Just, you know, get to something that works and let's go with this. You know, even a guy like Bob Langer, I've talked to him about this in the past. I've actually presented at his conference on this type of topic. And, you know, I got to say, I don't think everybody's going, hey, Chad said this, so let's go start making everything this way, right? There's not, it's not like there's you know, total belief that this is the case. But I think you have to believe that it's important. To me, it's just inconceivable that structure doesn't make a difference. And so, you know, we're going to go with that hypothesis and ask, can we begin to build systems that outperform what are you know, typically found via trial and error way? You know, what's a good stimulant? What's a good adjuvant? What's a good peptide choice? But if we're right, what's really interesting and I think profound is that you have to ask yourself, if we had a vaccine that was made of adjuvant 1 and antigen 2, did we have a bad vaccine, you know, didn't work, or did we have the wrong structure? So it makes you rethink, maybe if I take the components and structure them properly, something that was a poor performer could be a great performer. Because I showed you some spherical nuclei that had the exact same components that were, you know, lousy in terms of vaccine performance, and restructure them and they're much better. And so that's kind of the unnerving thing, because it means then you'd better be careful about discarding components as you know, good or bad components based upon studies that did not involve structure. And so we're a long way, I think, from before everybody fully accepts that or not. But I have a feeling that this is going to play a big role. And my feeling is reinforced by Michelle, who I mentioned, Toplensky, she has shots on goal now for COVID vaccines with the South African variant. So you can put these structures together very rapidly in active forms, and we're getting great data in animals. So that's coming down the pike as well. It's not just for cancer, but perhaps for infectious disease as well.
Great. Okay. So maybe another question for you, Christie, based on some things that different people have asked about. You know, when you were at the beginning of your talk, when you were talking about chemical modification systems for some of the hydrogels that you've been developing, and then you went into all of your light-induced stuff. I mean, one thing that I always think a lot about is that, you know, in biology, gradients are so important, right? It's not usually kind of off on. There's a gradient of activity, and then that may be what eventually creates a phenotype. So how do you guys think about that, just in terms of how well you can recapitulate those types of effects in your systems?
Yeah, no, that's a great question. And it's not just, oftentimes it's not just a single gradient, right? It might be opposing gradients that dominate a lot of biology. And so I guess one of the challenges, I think, for the field is knowing that you have, especially in the materials field, we have all different ways to track cells and their behavior, but it's really hard to prove. That's what you've done with the material, if that makes sense. So like gradients are tricky. Like, do I need to have a gradient over a cell or over entire tissue? And what scale and what, slope and things like that. And so that's, where we spend a lot of our time thinking about proving that we have what we think we have, and does it matter? But absolutely, gradients matter. It's not about single molecules, about multifunctional materials. Chad gave great examples of that too, clustering signals and things.
Absolutely. Great. Okay. Well, as I said, I do want to spend the last few minutes just on your perspectives on women in science. And, Chad, when you were starting your talk and you're talking about Hillary Godwin, I remember very vividly interviewing at Northwestern. I think you were already an assistant professor there. And a lot of the people that were touring me around the chemistry department said, no, we don't have any women faculty, but don't worry, we are about to get one. And I can't tell you how many people told me that during that visit. And I did think, wow, that's really incredible. Like, how could there be no women in this department? Obviously, that situation has resolved itself, there's been a lot of progress made. But I personally think a lot about how far we've come. I've seen many, many more women in science, it feels like, at least, than when I started out. But at the same time, in my day-to-day, I notice or think about the fact that we still have a ways to go, right? And there's so much more awareness. 20 years ago, we would not have really had an event where we were talking about things like this. So I think, the world has changed in so many wonderful ways, but, we have to keep pushing. So, Christie, maybe you could start just with your perspective on your trajectory, and I'm sure many situations being the only woman in the room, and maybe then seeing more women in the room, and then maybe we could have Chad, you close us out just with your perspectives. I know you've had a lot of wonderful female trainees that you've mentored and sent off to do great things, so maybe we could just do that as our final spin. So over to you, Christie.
Sure. I began my faculty career at the University of Colorado in 1996, and I was the only woman faculty member in my department. But I came here and I had amazing mentors. They happened to all be men. It was Nicholas Pepys, Bob Langer, and they were fantastic. And so I spent the early part of my career, I wanted to be recognized for being outstanding faculty and researcher and teacher, and do it, and not just the woman faculty member, woman. But I also realized it was quite an honor and privilege that the students that came through CU and that wanted to have someone that they could see themselves in, and to that they could appreciate, or they could envision someone and a career path that they could take. And so I realized that it was quite an honor and a privilege to be a mentor and be authentic and be someone that could inspire them and help them reach their career goals. And I also really appreciate being part of the Gordon Research Conferences. They have an amazing group of people, and I'm really inspired by what they do to try to support the diversity in our fields. And it's a great tribute to Nancy Gray and their whole team with what they do. It gives me lots of hope for where our future and our field is going.
Yeah, couldn't agree more. All right, Chad.
Yeah, so I mean, it's kind of interesting. As I said, I kind of lived through this, right? With going through the early days of Northwestern and the transition. People make good things happen. Hillary was somebody who really did. She took a chance on us, and it had a huge impact. And today we have incredibly accomplished female colleagues, and we're going to have a lot more as we continue to go down this path. But you look at it, you used to always, okay, Marie Curie, you mentioned the same person from a long time ago. You'd be like struggling to figure out who really contributes. To me, it's incredible. The last 2 Nobel Prizes have gone to women, right? Multiple women. It's an amazing thing. I mean, a lot of the real world leaders now women. That's an incredible transformation in a relatively short period of time. There still is a long way to go. There's no doubt about it. But if I look at my own group, I've never selected people based upon male or female. As I said, I've always just gone by interest, and I've had a large number of women that have come into the group, different times, and we've gone through ups and downs in that area. But I look at the people that are in the group now. We have 20 people now that are faculty at major institutions around the world. And just in this year's class, 5 women competing for top 25 jobs around the nation and getting them, which is exciting. Again, wouldn't have even crossed my mind that was a possibility 25 years ago. So I think signs are that we're moving in the right direction. I think that we have to pay more attention to increase in the pool. I don't mean the pool of faculty, I actually mean the pool of students. And I don't mean just the pool of students based upon sex. I mean the pool of students that want to go into this particular profession. I think that to me was probably the biggest kind of shock to me, that because I think of this as the greatest job anybody could ever have, that somebody would actually take themselves out of competition for one of these, that had the skills to do it. But a lot of women just didn't want to go into this profession in the 90s, at least in chemistry. And I think that's changed, at least based upon my own micro experiment at Northwestern. That's changed. We have an enormous number of women that are major leaders in the group and landing big positions, and I think are going to be the thought leaders of the field moving forward.
Absolutely. Well, I couldn't agree more with the comments that you've both made. Christie, I had those same wonderful mentors that helped me really launch my career, and now I can do the same for others. Chad, I know you've had a huge impact on your female trainees. I know some of them personally that have told me about how you've supported them and then almost maybe pushed them towards academia because you knew they were cut out for it. And I think that is how we really move the needle. So I want to thank you both again for great talks, really insightful discussions. Great to see both of you. I want to thank Nancy for inviting me to be the moderator today, and I will turn it back to you.
Thank you. I want to thank Christie and Chad for phenomenal talks. So interesting and accessible and important. So thank you so much for volunteering your time, and Shana for serving as moderator and leading a great discussion. I also want to thank Christie and Shana for serving as true role models of women in science, and for Chad being such a tremendous advocate for women in science. It means a tremendous amount to all of us. So thank you again. And we're going to close with a short video. about how you can provide feedback on our event today and how you can participate in future GRC Connects events. So thank you again. Happy International Women's Day, and we hope to see you soon. Bye.
Thank you all for attending today's event. Very shortly, you will receive a brief survey. Please provide feedback so we can continue to plan future GRC Connects events. In addition to the feedback survey, you will have the opportunity to contribute to one of GRC's diversity initiatives. This helps us attain our goal of holding open, diverse, and accessible meetings. We want to thank those who have made a contribution prior to this event. We greatly appreciate all of your support. So if you or your community are interested in hosting a GRC Connects event of your own, please reach out through connects@grc.org. And don't forget to follow us on all social media platforms so we can stay connected. We're not sure exactly when we will be seeing you, but rest assured our communities will come back stronger, more united, and more excited than ever before to advance the frontiers of science. Thank you all for attending today's event, and thank you to our incredible speakers. We hope to see you all very soon.